Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, right. So we're currently in the process of discussing smooth muscle cell contraction as an example of IP3 mediated calcium signaling. Okay, right. So we've now discussed how when a calcium signal occurs within a smooth muscle cell, how a global calcium signal in a smooth muscle cell can trigger the initiation of contraction in that smooth muscle cell. We've discussed that calcium acting through calcium calmodulin complexes can activate the myosin light chain kinase, which will then phosphorylate the regulatory myosin light chains of a myosin monomer, within the myosin filaments. And those myosin monomers are now going to be able to actually bind to the myosin binding sites of actin monomers in actin filaments. We've also discussed how calcium calmodulin complexes are going to bind to calponin proteins, cause that change in conformation in calponin proteins, which are then going to lead to the tropomyosin proteins being reoriented so that they're no longer covering up the myosin binding sites of the actin monomers in the actin filaments. Okay, so that's how we get the contractile machinery actually ready to initiate the contraction process. But we haven't yet actually discussed the contraction process itself. Okay, we haven't actually discussed how the myosin heads of myosin filaments actually interact with these myosin binding sites of actin monomers in actin filaments and trigger the movement of the actin filaments, and that's what we now want to study. Okay, and this is a process called Crossbridge cycling. Okay, right. We are going to go through the process of crossbridge cycling, even though we have now actually done the bit which calcium plays its part in. Okay, calcium is just involved in initiating the ability for crossbridge cycling to occur. Okay, it doesn't actually play a part in the actual crossbridge cycling itself. However, because muscle contraction is such a central example of calcium signaling, and indeed we're going to look later at skeletal and cardiac muscle contraction as well, I feel that I should include a discussion of the crossbridge cycling process, okay, so that you have a complete picture of muscle contraction. Right, okay, so what I'm going to do is I'm going to show my actin filaments slightly simpler than I have previously been showing them. So we've been talking about how actin filaments consist of these two F-actins which are intertwined around one another. However, it's awfully complicated to continuously draw it intertwined around one another. So the way I'm now going to show my actin filaments is just as with the two F-actins sitting parallel to one another but not intertwined around one another. That's not correct, okay? They are intertwined around one another. However, it's just easier to draw them not intertwined around one another. So I'm going to go over to showing my actin filament like so. Okay, so I now want to show you how a myosin head can interact with the actin filament. Now, before we even begin this process, I just want to make sure that one point is absolutely clear. Okay, when myosin filaments have an effect on an actin filament here, okay, when they pull the actin filament along, you have to remember that that is the combined effort of a huge number of myosin heads interacting with this actin filament. Okay, so it's not just a single crossbridge cycling event involving a single myosin head that is doing this movement. Loads of them are working at the same time, basically, and that's what triggers this overall movement of the myosin molecules of the actin filaments by the myosin filaments. Okay, so it's important to be aware of that. Okay, and I will come back and note that point at several points in our discussion. Okay, so here then is our actin filament, okay, or a piece of our actin filament at least, and now I'm going to put our myosin monomer, okay, and this will be a myosin monomer within a myosin filament, but for simplicity I'm just going to show a single myosin monomer here. Okay, so here is a myosin monomer within a myosin filament. Now you can see the direction in which this myosin monomer is oriented. Okay, its tail is facing that way, so that means that it's going to be helping to pull this actin filament in this direction here. Okay, towards the tail of the myosin monomer. Okay, so I'll colour in this myosin monomer here in blue. Okay, right. So, even before the calcium signal has occurred, the head of the myosin monomer has two molecules bound to it. Okay, it has an ADP molecule bound to it, okay, which I'll show here, and it also has a phosphate group bound to it. And I want to stress that this phosphate group is completely different from the phosphate groups that the myosin light chain kinase enzyme will add on to the regulatory myosin light chains of the myosin head. Okay, so this is present even before you have had the calcium signal, and even before the myosin light chain kinase has added those phosphate groups onto the regulatory myosin light chains. Okay, so don't confuse that phosphate group with the phosphate groups that the myosin light chain kinase will add on to the two regulatory myosin light chains of the myosin monomer head here. Okay, right, so here are these ADP and inorganic phosphate molecules bound to the myosin monomer's head then. Okay, now, what will then happen is you'll get a calcium signal occurring. The calcium will result in the phosphorylation of the regulatory myosin light chains within the myosin monomer's head. Okay, it will also result in the removal of the tropomyosin block of the actin monomer's myosin binding sites. So now what's going to happen is the myosin head of this myosin monomer can bind to the myosin binding site of one of these actin monomers in this actin filament here. Okay, so let me just show this. So now, once again, here is our actin filament shown like so. Okay, and now we have formed an interaction between the myosin monomer's head and one of the actin monomers in this actin filament here. Okay, and at present, the myosin head will still have the ADP and the inorganic phosphate attached to it, at least transiently. Okay, so I'll show this transient intermediate stage here. Okay, so once again I'll colour code this in. So here are the actin monomers of this actin filament, which we'll have in green here. Okay, then we'll colour in the myosin monomer here in blue. Okay, we'll colour the ADP here once again in orange. We'll colour in the inorganic phosphate group here in purple. Okay, so we've formed this interaction then between the myosin head and the actin monomer. Okay, and this bond is called a crossbridge. Okay, so we have formed the crossbridge at the moment. Okay, right, now, I said this was a very transient stage, what I've drawn out, and indeed it is, because the moment the myosin head binds to an actin monomer within the actin filament, what's going to happen is that phosphate group that it had bound to it is going to be released. So this is going to be released. Okay, and when that phosphate group is released from the myosin monomer's head, what happens is the myosin monomer's head changes conformation. Okay, so at present, the way I've shown the myosin monomer's head, it's sort of quite straight, okay? It's quite tall and pointing upwards, okay? What's going to happen is the moment it releases that phosphate group, it's going to change conformation. It's going to go from being pointing straight upwards to pointing more at a 45-degree angle like that, okay? And when it does so, it's going to end up dragging the actin filament with it. Okay, and this movement is known as the power stroke of the myosin head basically, and this is the portion that is actually going to move the actin filament, or at least contribute to the moving of the actin filament. So you have to remember there are going to be lots of myosin monomers all doing this on this same actin filament, and they're all going to be contributing their power strokes to trying to move this actin filament. Okay, so let me now show you then what state this will be in after the power stroke has actually occurred. Okay, so once again I'll start by drawing my actin filament out again here. Okay, and now the myosin monomer's head will be at this sort of an angle instead of the previous angle that we've seen it at. Okay, and it's important to understand it's not the hinge that has changed, it's the head that has changed. The head has changed its orientation. Okay, and now of course the phosphate group is gone basically, but you still do have that ADP molecule there. Okay, right, so let's colour in this picture. So we'll colour in the myosin monomer here in blue. Okay, we'll colour in the actin monomers once again in green here. Okay, and then we'll colour in the ADP molecule in orange. Okay, right, so that is what we've got after the power stroke, and I'll stress again, the power stroke is how you actually generate some force, how you actually generate some movement of that actin filament. Okay, and I'll once again stress that the myosin filament will have loads of myosin monomers with loads of myosin heads, all attaching to the same actin filament and all performing power strokes basically on the, all attaching to this actin filament and all performing power strokes on that actin filament to try and move the actin filament in the direction that it wants it to move. Okay, right. Now what's going to happen is the ADP molecule is going to cleave off. Now that doesn't do anything. All that does is makes available a new binding site that a fresh molecule of ATP can now come into and bind to. Okay, so now what's going to happen is a fresh molecule here of ATP is going to come in and replace that molecule of ADP that was bound there. Okay, so in now comes the ATP. It's going to bind where the ADP used to be bound, and when the ATP binds to the myosin monomer's head, it will then cause the cleavage of the crossbridge, basically. Okay, so now what you're going to get is that crossbridge is going to break apart. Okay, so once again, here is my actin filament shown here. Okay, now I have cleaved the crossbridge apart. Okay, like so. Here's my myosin monomer here. Okay, and now I've got ATP bound to the head of my myosin monomer instead now. Okay, so the ATP has caused the crossbridge to cleave. Okay, now you might ask, well, why doesn't the actin filament now just spring back to its original position? Okay, but then again, it's another point where it's important to understand that there is not just going to be one myosin head attaching to the actin filament. The reason the actin filament doesn't just spring back to its original position is because there are loads of other myosin heads of the myosin filament attached to the actin filament and holding it in position basically. So this one breaking its crossbridge isn't going to allow the actin filament to move back because loads of others will still have their crossbridges and will be holding it in the position that it currently is in. Okay, so if you want the actin filament to spring back, basically, you have to break all of the cross bridges between all of the myosin heads of the myosin filament and the actin filament. And that's what will happen when we come to relax the smooth muscle cell back down. We'll have to break all of the cross bridges. Okay, then what's going to happen is you're going to hydrolyze that ATP molecule down to an ADP and an inorganic phosphate, and that will cause the myosin head to return back to its original conformation. Okay, so once again, I'll show the actin filament here, like so, and now this is going to change conformation now again. Okay, it's going to be sitting tall again, straight up, like so. Okay, and now it's going to have an ADP molecule and an inorganic phosphate attached to it. So it's back to square one basically. However, you will notice that it has moved along the actin filament. Okay, it's now in the same state as it was originally. Okay, so if I colour things in, it'll become more obvious. Here is our myosin monomer here. Okay, and now again it's back at square one where it has ADP bound to it and inorganic phosphate bound to it. Okay, however, you will notice that its position relative to the actin filament has changed. Okay, the tip of the head is now next to this actin monomer here rather than this one here. Okay, so it has moved up 2 actin monomers basically, and that is indeed how many it moves up when it does one of these cross-bridge cycles. It moves up 2 actin monomers, and now it can do the same thing. It can go through the exact same process with this actin monomer here, and it can continue to move the actin filament in this direction. So basically the myosin filaments, or rather the myosin monomers of the myosin filaments, climb up the actin filament basically, and they're all doing it. So, you know, the myosin filament will have a huge number of myosin monomers within it. So you'll have a huge number of myosin heads sticking out from the central fibre of the myosin filament, and they'll all be climbing up the actin filaments around them, and that's how you end up getting this pulling of the actin filaments towards the centre of the myosin filaments here. Okay, right, so that then is the sliding filament mechanism of muscle contraction, or the mechanism of cross-bridge cycling. Okay, what I just want to talk about is what happens then if you want to relax the smooth muscle cell back down. Okay, so if the smooth muscle cell wants to relax back down, then the calcium signal needs to go, which of course it will if we're having calcium waves propagating across the smooth muscle cell. So what will be happening is you'll continuously get calcium rising and then going back down. So when the calcium goes back down, the smooth muscle cell will then relax back down. So what happens is once the calcium goes down, what's going to happen is calcium calmodulin complexes will go down in the cytoplasm of the cell. You'll therefore stop activating myosin light chain kinase. Okay, so myosin light chain kinase will stop adding phosphate groups onto regulatory myosin light chains of myosin monomers in myosin filaments. Then there is another enzyme which will now remove the phosphate groups from the regulatory myosin light chains. Okay, and this is called the MLCP enzyme, which is the myosin light chain phosphatase enzyme. Okay, so this enzyme is active in the cytoplasm, and it will remove the phosphate groups from the regulatory myosin light chains. Now it's normally functional, okay, it will have been removing phosphate groups from regulatory myosin light chains all the time through whilst you've been contracting. Okay, the difference will be that when you were actually contracting and you had the calcium signal, the myosin light chain kinase was adding the phosphate groups back on all the time, and therefore it didn't matter that the myosin light chain phosphatase was removing them. Okay, but of course once the myosin light chain kinase is no longer actually functioning because the calcium signal is gone, then myosin light chain phosphatase will dominate. It will start removing all the phosphate groups from the regulatory myosin light chains, and they're not now going to be replaced anymore, so the myosin monomers will therefore lose their ability to bind to the myosin binding sites of actin monomers in the actin filaments. So that will start to mean that cross bridges will be cleaved, but then new cross bridges will not be formed. In addition, the calcium calmodulin complexes will cleave off the calponin molecules, okay, as the calcium becomes detached from the calmodulin molecules, okay. That will cause the reorientation of the tropomyosin over the myosin binding sites of the actin monomers in the actin filaments, okay, which will also start to block myosin heads from reforming cross bridges. So basically you're going to be getting cross bridges between the myosin molecules of the myosin filament and the actin monomers of the actin filaments cleaving all over the place basically. And once all of the cross bridges have been broken, then what will happen is the actin filaments will then recoil back out. They'll go back to their original position, but that's really important to understand. If you want the actin filaments to recoil back out, then you need to break all of the cross bridges, not just one or two, they all need to go basically, so that the actin filaments are effectively let go of and they can return to their original position. Okay, and that will lead to the entire smooth muscle cell returning to its original size. Okay, right, so that is the sliding filament mechanism of muscle contraction, or cross-bridge cycling. Okay, what we will turn our attention onto in the next video is we will move on to ryanodine receptors and their involvement in releasing calcium from intracellular stores, particularly in skeletal and cardiac muscle cells.